Ribosome is a pretty fantastic system, and it really lets us get some clues about evolution before Luca, but I'd just like to let you know it's not the only game in town. There are other RNAs that we can look at that were present in Luca, not very many, but one of them is a nuclease, and for those of you who know a lot about RNA, you probably already guessed that I'm talking about Ribonuclease P. Ribonuclease P is a nearly universal ribozyme. That is to say, there are a few organisms scattered here and there around the tree of life that have dispensed with the RNA component of RNase P, but by and large, pretty much everything uses an RNase P RNA to process transfer RNAs. So the main thing RNase P does is it takes a pre-tRNA, cuts off a sequence from the 5' end, and gives you a mature tRNA. They can go on and do its job with the ribosome and make proteins, okay? All right, so that's what it does. As I said, it is present in all three domains of life. Here are examples from bacteria, archaea, and eukaryotes. And as you can see, they all have different secondary structures, but there are certain elements that are shared between all three of them. And so they have certain sequence elements in common, they have certain structural elements in common, and they're widely dispersed across all three domains, so it's quite clear that this was present in Luca. And in fact, one other interesting thing about RNase P is that it's a ribozyme, which there are not that many of. And it's true in biology. RNase P is always has at least one protein component, but it's not necessary for catalysis. So this is a ribozyme, and because it doesn't need its protein, and it's universally conserved, and it was in Luca, I like to point out that I wouldn't say that RNase P is older than a ribosome, but it is something that could be older than the ribosome, and it's possibly the only other system where we could have follow a continuous evolutionary track from before the ribosome to now. Okay, so it's pretty cool. We should learn more about it. So there were a bunch of talks about in the last session about the accretion model. So I won't go into too many details, but it's a model developed by largely by my collaborators at Georgia Tech, and it explains ribosome evolution. And there are sort of three key features, as far as I can tell from talking to them and reading their papers, and that is that. One of the key features of this model is that structural RNAs, if you look at an RNA structure, you can see a record of where elements were inserted into it. That if you look at a three-dimensional structure, you can also get information about the order in which elements were added, and that these RNAs generally grow from a common core. And so that's the model with the ribosome, so why not apply it to RNase P? This model relies on three-dimensional structures from different domains of life, and now for RNase P, as of about a week ago, we have bacterial, eukaryote, and this just came out, archaeal RNase P structure. And so we have high-resolution three-dimensional structural information for all three domains of life. And so I just want to go through a couple of examples of some of the features of the accretion model as they show up in RNase P. So this is P18. It's one of the helices in RNase P from bacterial structure. And what I'm showing is what we consider an insertion site. That is a site where it looks like you could have put an element into the RNA structure without disrupting the rest of the structure, and that is to say this part that's shown in cyan, we think was dropped into the part that's shown in blue. And you might think, okay, yeah, well, the backbone, it comes together. Just because the backbone's coming close together doesn't mean that it used to be connected. But if we overlay it with the archaeal RNase P structure, then we see that, you basically have the same structure minus this P18. And of course, the other thing about that, there are other reasons we kind of know this element is probably a later addition. It's not in eukaryotes or archaea, and it's not even in all bacteria. So, but at any rate, we can see some of those features in RNase P like we see in the ribosome. Another thing that we see in the ribosome a lot of are these A minor interactions. They're also present in RNase P. And so what's going on there is that if you look, you can see in blue we have a double-stranded RNA. That double-stranded RNA can form on its own. It doesn't need the part that's shown in cyan and in green. Okay, the part that's shown in green, these are a couple of adenosines that are looped out and they're interacting with the minor groove of the double-stranded RNA. That structure will not form if the double-stranded RNA is not there. And so the thinking is that the independent structure is older than the dependent structure. Okay, So we have those two elements. We can find insertion sites, and we can establish chronology based on three-dimensional structure. So then if we go on from there and we kind of build up the structure from a single core, we can kind of have a model of how RNase P evolved. And so what I'm showing here is RNase P substrate. It's a tRNA. There we go. All right, so basically this part right here kind of shown in gray is the piece that's going to get cut off. There are a couple of metals at the active site where the cleavage happens, and we'll just kind of walk through the evolution of RNase P. So we start to have different chunks get added on. After the first four pieces are added, we have basically have everything that you should need for catalysis. And then as you continue on in the model with through time, you add on additional elements that recognize larger and larger portions of the tRNA and also help to stabilize some of the structure. And then we can go beyond Luca with this particular structure. There are a couple of additional elements that are specific to this lineage. Okay, so that's the fun movie. Here's a couple other views. How am I on time?
Checking.
Okay.
4 minutes.
Oh, okay. Oh, great. Plenty of time. Okay. So here's a couple of different views of the model and broken up in different pieces. So what do we do with this now that we have this model? Well, there are a couple things we can do, and one of the first things I wanted to do was sort of track how does the interface between RNase P and its substrate change over time. So we start here on the left. Yes, very early on. And so some of the first things that are contacted are the cleavage site. That's not surprising. And then there's also part of the tRNA that's one helical turn away from the cleavage site. And then as we go forward in history, we start to contact more of the leader sequence, the part that we cut off, and then even more of the acceptor stem, then the T-stem T-loop, and then the last thing that gets contacted is the D-loop. And what's kind of interesting there is that it's not really until you get to Luca that you start interacting with parts of the tRNA that are sort of unique to tRNA. So obviously the acceptor stem at the ends is unique. It has a CCA tail and all that, but it's essentially interacting with what you might call a mini helix up until Luca. Potentially. So that suggests that, maybe the full tRNA didn't come till later. Okay, so, and just to, I don't really have, well, this is my last slide, and I'll just say that we're kind of right in the thick of it and like figuring out this model and what it all means. But one of the things that occurred to me is that by tracking the way this interacts with tRNA, we might be able to synchronize the evolution of RNase P with the evolution of the ribosome, with the idea that some of the same that the ribosome and RNase P might be contacting parts of the tRNA at the same time. And so, like I said before, for RNase P, we don't contact the D-loop until very late, and the ribosome doesn't start to really interact with the D-loop either until fairly late. So something along there. We've got a lot of work to do to really flesh out this model and make sure it's well synchronized with the ribosome. This is definitely one of those projects where I'm talking about it as I'm thinking about it, and so we don't have a nicely tied up package for what it all means. But I think this is a very important system. It has a lot of potential. I haven't heard a lot of talk about it at Origin of Life or Astrobiology conferences in a while, and so I think we need to revisit it, especially now that we have all this structural information. And with that, I'd just like to thank my collaborators at Georgia Tech, especially Anton, who I think it was last night or the night before, we spent several hours looking at structures, having a lot of fun. This work was done, I'm at NASA, they're at Georgia Tech, and I thank you for your attention, and I hope we have a little time for questions. Okay.
You want a microphone?
Is the protein ancient, or what can you say about the protein?
Well, you can say that it's totally dispensable. It looks pretty. It's it's fairly sophisticated. Right? It's mixed alpha helices and beta sheets. It doesn't really match what some of the accretion models or the Williams Labs model on early proteins, and it seems to be a lot of what's involved is recognizing the 5' leader it does help catalysis, but it's primarily sort of a recognition thing. And I should point out that the other RNase, so that's bacterial RNase P, just has this one protein that's shown in brown here over on the left, but archaeal and eukaryotic RNase Ps have several more proteins. Eukaryotes have up to 10 or so, and half a dozen for archaea. So, and what's kind of interesting is in some cases you can kind of see deletions that happen in eukaryotes or archaea, where you kind of have a protein that seems to be coming in and compensating for, or taking the place of an RNA element.
Anyway, cool stuff. But I was taken when you showed the 3 ribonuclease Ps. 2 of them, one from Chaldococcus yonashii, and the other are thermophilic. Is there any implication from what you're doing that perhaps the first ribonuclease P was high temperature, and that whether or not that might actually fit with some of the models that indicate that Luca was also a high temperature growing?
Well, I'll just say that the examples I showed were the examples for which we have 3-dimensional structures, and so it really just speaks to the fact that the thermophiles are easier to get structural information on. Some of the RNAs associated with thermophiles tend to be,
But you don't have a model that indicates what the first ribonuclease P would have looked like.
Well, we do. I mean, as the movie, we kind of march through, and we haven't done. I mean, what's nice about the system is it's fairly small, and we can go and characterize the activity of some of these constructs. So all those different steps, we plan to make in the lab, and it won't be that difficult to make. It's not a very large RNA, and we know that a lot of truncated forms of RNase P are catalytically active. You can cut off tremendous amounts of RNase P and still have activity. So we're very optimistic that as we build constructs that correspond to the different steps in the accretion model, that we'll be able to test them and characterize, what temperature do they work best at, what pH, what ions, how they respond to iron, things like that.
Yeah, my only comment is that I was really taken by the fact that you have ribonuclease P functional at above 90 degrees in yonashii, for example. So I'm very interested in how that is different from ribonuclease P and E. coli, for example.
Oh, okay.
Okay. Thank you, Mark.